webinar. Thank you for joining us in this CureDuchenne webinar series. So CureDuchenne, as many of you know, fund a tremendous amount of research. And over the past couple of years, I've come across a few companies and scientists who have some really innovative research that they would like to try on Duchenne that looks very, very promising in other indications. But these scientists could not find biospecimens to test their therapies on. And it's been a really frustrating journey to try and help them secure biosamples so that they can do research. And and there's another issue that comes up often also, that the samples that are available tend to be centered on the most prominent and prevalent mutations, the ones that are being tested right now, meaning exon 51 amenable mutations, exon 53. And once you get past those particular biosamples, they're few and far between. And for me, you know, as a parent and also as an advocacy leader and also as a funder of science, we find that very unacceptable. Right now, only 5% of rare diseases have a treatment approved by the FDA. And to a certain extent, this is due because of the lack of quality biospecimens for research. And so at CureDuchenne, we are committed to removing any barrier to a cure. To fill this gap, CureDuchenne has initiated the CureDuchenne Biobank, which is a collection of biological material and data from Duchenne patients stored in an organized system that is accessible to all. And we've partnered with Rutgers Infinite Biologics for the Biobank services, and also with UCI Irvine here in Irvine, California. And I'm going to introduce two esteemed scientists here and physicians that are partnering with us on this biobank. We have a board of experts in biorepositories and registries, technology transfer, genetics, neuromuscular disease, immunology, and ethical and legal and social issues. I'm going to be introducing Dr. Tassine Mozaffar and Dr. Armando Villalta. But first, I'd like to remind everybody that we will be taking questions, and you can hover your mouse over the lower part of your screen and click on the Q&A icon, and you can type in your questions, and they will be answered at the end of the webinar. This is a short webinar, only going for a half hour. And so with that, I'm going to introduce Dr. Tassine Mozaffar, who is professor and interim chair of neurology, interim neurology chair at the Institutional Review Board at the University of Irvine, California. And Dr. Armando Villalta, who is assistant professor of physiology and biophysics and the School of Medicine at University of California, Irvine also. So with that, I'm going to pass the presentation over to Dr. Mozaffar.
Thank you, Deborah. It's a real pleasure to be able to participate in this webinar. I have been associated with CureDuchenne for many, many, many years. When I first met Deborah almost 17 years ago. But this is a great opportunity for us to work together along with Dr. Villalta to really develop a high quality biospecimen collections. My team, including some of the physicians that work with me, as well as some of our research coordinators, are helping collect some of this biospecimens for future use by national as well as international scientists to do research in Duchenne muscular dystrophy. So the purpose is for us as the UCI clinical team to collect minimally invasive samples. This mainly constitutes of blood samples and skin tissue samples. And within blood samples, we are collecting materials for immunological studies through what's called PBMCs, as well as DNA and RNA samples in a separate blood tube. And the skin tissues are taken so they can be transformed into fibroblasts. And then eventually we can do all sorts of studies, studying the gene mutation, see that if that mutation can be edited through some of the newer biotechnology that are now available. It's almost impossible to do any of this research without looking at healthy controls. And these healthy controls are either first degree relatives of the patient with Duchenne muscular dystrophy or age match control. So as part of this collection, we are encouraging parents as well as siblings, but also age matched individuals who are between the ages of 3 and 24 to give their blood samples for comparative studies as well. Now, from these skin biopsies, we will be able to collect personalized cell lines. These are cell lines that are going to be specific to each patient, and they will also carry the same mutation that the patient has. So we can understand the effect of these mutation on the disease further. But as I mentioned before, we have the opportunity to correct them in the cell line as a proof of concept whether these mutations are correctable through gene editing and some of the other technology. But these cell lines also provide us an opportunity to test the different FDA-approved medications that are already on the market in a cell culture model, so we can pick out the best treatment paradigms for Duchenne muscular dystrophy. So it's really reduces the amount of time that it takes to bring a drug to clinical stage. Now, as I've already alluded to this, but again, as I mentioned, the cell lines are going to be unique to patients' genetics. They will have the same mutation that the patient has. And it also allows us to develop various biomarkers. These are markers of disease that can be used to study disease progression or disease response to treatment. So having a unique collection of blood and skin samples from these patients, which are very well characterized. We collect a series of clinical data as well as mutation data, as well as medication data that provides a unique opportunity to further progress in research and therapeutics in Duchenne muscular dystrophy. I'm going to hand over at this point to Dr. Vialta, and since he has a special interest in immunology, and he can describe to us what we will do with some of the samples that we collect.
Dr. Mozaffar, and thanks to CureDuchenne for including me in this. This is a really exciting time in participating in the development of this biobank. So in this last point here, but one of the things that we're very much interested in the field is personalized immunology. And part of the interest that kicks this off is what we've learned through a variety of studies that have been done in mouse or animal models of Duchenne, showing that the immune system has a really important role in the pathogenesis of Duchenne. With the advent of different gene therapies, micro and mini dystrophin, we also face similar questions when we think about exon skipping and really any strategy that aims at restoring dystrophin, how there's a potential autoimmune-like response that may be generated to the dystrophin that is restored in these type of therapies. So it becomes really critical at trying to understand the potential barrier that the immune system itself might act in these type of therapies. We also know from other diseases in which AAV has been used as a delivery system for therapeutic genes in those settings that the AAV vector itself is immunogenic and can be perceived as a foreign pathogen. So there's also limitations there that we as a field need to address. As one of the things that we've also become very interested in the immune system outside of the potential immunogenicity in the context of gene therapy is also understanding the role of the immune system in tissue repair and regeneration, which there's a lot of studies that have been done in animal models that support this. And this is certainly the case for skeletal muscle. We've learned that there is a huge role for the role of the immune system in promoting regeneration. But one of the limitations is that we don't know whether many of these things are actually occurring in human. And part of the challenge that is accessing patient samples to try to understand the parallels that exist between mouse and human. We have, however, through animal studies, determined that in addition to the role of the immune system in regeneration, that in chronic situations like Duchenne muscular dystrophy, there's a broad dysregulation of the immune system where these normal inflammatory responses become defective. So the animal studies have been very informative in this regard, but we're meeting a barrier that has limited our ability to translate the information that we're learning from animal studies into human. And again, as I mentioned before, that this has largely been attributed to our inability to access human samples, as Deborah was mentioning at the beginning of this webinar. So this becomes a point where the community as a whole, family and patients, become really critical at meeting or breaking down that barrier. And that's through the donation of precious biospecimens, which could be in the form of blood, skin biopsies, or even muscle biopsies that will be really informative for the scientific and medical community in developing new therapies that are aimed at targeting the immune system. And I can speak from firsthand. Dr. Mozafar and I have been working for a few years together now. And I recently met the folks at CureDuchenne, and I can tell you that this effort has been super impressive. And we recently acquired samples through this effort, and we're currently in the lab analyzing data from experiments. So it's going to be a really exciting period with this as a huge resource for the field that's going to really push the research forward.
All right, thank you, Armando. This is Romina Foster-Bonds. I am the Director of Programs at CureDuchenne. Thank you so much for joining us today. We are so pleased of the interest from our community. So any questions about the biobank, please feel free to reach me directly. I am working on scheduling future collections to meet the demand, so please stay tuned for updates on new biobank collections that will be done alongside our CureDuchenne events across the country. So lastly, I want to end with the opportunity to ask any questions. And we will be submitting a survey tomorrow morning. So any questions that weren't submitted today, you can please ask your questions through the survey, and we will work on answering those questions and actually archiving the answers to those questions alongside with our webinar on our website. So I don't see that any questions came in, so please submit your questions in the post-webinar survey or reach out to me if anything else is needed. Thank you so much for joining us, and have a wonderful rest of your day.